Looking at this busy market street in Kampala, the sheer volume of sensory information is staggering. Within this environment, your attention immediately snaps to a specific mechanical stimulus, the sudden, loud hooting of an approaching boda-boda. Look across the street, and you pick up a distinct optical signal, sunlight bouncing off a vendor's bright yellow bananas. But there is a physiological catch. Your brain sits locked inside a dark, completely silent vault of bone. It has no direct contact with those bananas or that motorcycle. To interact with the outside world, your body relies on receptor organs. These structures act as active transducers, capturing raw environmental data and converting it into the brain's singular language, electrical impulses. The two most complex systems performing this conversion are the eye, which utilizes photoreceptors, and the ear, which relies on mechanoreceptors. On the left, a photon of light; on the right, a sound wave. Despite their physical differences, both convert into identical electrical sparks. Sensory organs function as specialized data processors, translating physical environmental energy into the electrical information required for neural perception. In this classroom, light reflects off the chalkboard, carrying the visual information of the lesson toward the students. This cross-section maps the path of that light. The first structure it encounters is the cornea, the clear front window. The cornea is responsible for the primary bending or refraction of incoming light. The light then passes through the pupil. The iris provides muscular control over this aperture, physically contracting to shrink the pupil in bright sunlight or widening it in the dark. Next, the light reaches the lens. This flexible biconvex structure fine-tunes the refraction, causing the incoming light rays to angle sharply inward and cross each other. Those crossing rays strike the retina at the back of the eye. Acting like a biological projector screen, this layer of millions of rods and cones captures the light as a focused, inverted image. The visual data exits the eye through the optic nerve. Because this juncture requires a physical gap in the photoreceptor layer, it creates a natural blind spot in our field of vision. Our two forward-facing eyes provide overlapping fields of vision, a geometry that allows the brain to calculate depth and distance. Every anatomical structure in the eye exists to manipulate optical physics, ensuring scattered environmental light converges into one sharp point on the retina. Clinical short-sightedness is a matter of strict anatomical geometry rather than disease or nutrition. Functionally, this condition is myopia. A student can clearly read a textbook on their desk, but the writing on the chalkboard remains blurry. This diagram reveals the anatomical cause of myopia. The eyeball is physically too long from front to back. Because of this elongation, the light rays cross too early. They form a focused image in the empty space in front of the retina, leaving only a blurry projection by the time the light hits the back wall. We correct this by placing a concave, or diverging lens in front of the eye. This lens spreads the incoming rays outward, delaying their intersection, so the final focal point falls directly onto the retina. A simple mnemonic for this correction is mycave. Myopia requires a concave lens. The opposite condition is hypermetropia, or long-sightedness. Here, distant objects are sharp, but near objects, like a newspaper at reading distance, are blurry. In this case, the anatomical cause is a shortened eyeball. The light rays reach the retina before they have a chance to cross, attempting to form an image behind the eye. To correct this, we use a convex, or converging lens. This pre-bends the light inward, pulling the focal crossover point forward until it rests precisely on the retina. Visual defects are structural geometry errors that can be perfectly offset by the application of corrective optical physics. The body's mechanoreceptors are located in the ear, which manages both acoustic energy for hearing and fluid dynamics for physical balance. This cross-section traces the acoustic pathway. The outer flap of flesh, the pinna, collects airborne sound waves and funnels them down the ear canal. These waves strike the tympanic membrane, or eardrum, converting acoustic energy into physical vibration. This vibration moves the ossicles, the three smallest bones in the body. They act as mechanical levers, amplifying the force of the sound wave as it passes through the middle ear. That force strikes the fluid-filled cochlea. The resulting ripple in the fluid causes tiny internal sensory hairs to bend sharply. The bending of these hairs is the exact moment mechanical force is converted into electrical impulses, which then travel up the auditory nerve to the brain. Just above the cochlea sit the semicircular canals. The fluid inside these loop-like structures shifts as you move your head, allowing your brain to calculate and maintain your physical balance. The ear functions as a multi-stage physical processor that gathering environmental force and mechanically amplifying it to trigger a neuroelectrical response. While the architectures of the eye and the ear differ, they share a singular fundamental purpose. The cornea and lens are specialized to refract photons of light, while the eardrum and ossicles are built to capture and leverage mechanical air pressure. Despite tracking different environmental stimuli, both pathways deliver the same biological currency, an electrical impulse sent via a nerve to the brain. Navigating a complex landscape like this requires the continuous coordination of these internal physiological processes, allowing us to interact with our surroundings safely. Understanding the physics of our sensory organs provides insight into the hardware of human perception, allowing us to process, protect, and mechanically correct our experience of the world.